does, but it increases due to this activity of this instructor neuron, is it phosphorylates the potassium channels and makes them close. And if you close potassium channels, you actually make the action potentials get longer, right? Because the cell won't repolarize so quickly. You make the action potential longer. When it gets down to the terminal, more calcium comes into the terminal, more transmitter is released, the synapse is stronger. So in this particular case, the instructor neuron works by basically turning off potassium channels in the terminal of the presynaptic cell. That makes the action potential longer, that makes the synaptic strength go up. Does the logic of this make sense? It's a beautiful story, and Eric Kandel, who's one of the physiologists who studied this preparation, several years ago won the Nobel Prize for his studies in aplysia, because this is one of the first preparations in which the cellular and molecular basis of learning was described. So again, more calcium atoms are spiked, more transmitter is released. So learning occurs in this case presynaptically by spike prolongation, making the action potential of the presynaptic neuron larger. Okay, let's look at another example. This is another example that actually I used to work on at a point in my postdoctoral career. It's another one, a case where you have a synapse between two cells, the granule cell and the Purkinje cell, and there's an instructor neuron that changes things called the climbing fiber. And this circuit is in our cerebellum. The cerebellum is a very, very important part of our brain, which is believed to be involved in certain procedural memory. The learning of fine motor skills is an important part of the cerebellum. In fact, there are more neurons in the cerebellum than any other region of our brain. There's little tiny cells called granule cells, and they actually play a part in the story. So the cerebellum has these really beautiful cells called Purkinje cells, and David's shown you many pictures of these through the years. They're sort of, as David said, they're like mohawk cells. So they have these beautiful dendrites in arbor that are basically two-dimensional, and they're all sort of stacked up next to one another. But they receive synapses from granule cells that sort of rise through the cerebellum and then bifurcate into branches that run this way. So you imagine like Purkinje cell, Purkinje cell, Purkinje cell, Purkinje, whole array of Purkinje cells, and the granule cells run. I'm sorry, you're getting your laser. But the granule cells that are run through, making synapses with a bunch of. My graduate students are laughing at me. My postdocs are laughing at me because I just almost blinded somebody. The granule cells will make contact with many, many, many Purkinje cells, and it's that synapse between the granule cells and the Purkinje cells that is plastic over time and exhibits not a prolonged increase in synaptic activity, not long-term differentiation, but long-term depression. So the parallel fibers basically are the axons of the granule cells, and they make these synapses with the Purkinje cells. Each Purkinje cell is connected by this climbing fiber that comes up and wraps around the cell body and all the dendrites of the Purkinje cell. And this is sort of the instructor cell in this particular circuit. So here's what an experiment in this preparation looks like.